research other experiments with the same frog that has been flown on, like I said, a Mach 6 they tried, and they also tried a simulator of microgravity, and they found different developments like enlarged brain and arched back, which activin is involved with the goosecoid gene, which is in charge of mesodermal development, which is the brain and spinal cord. So that's why we wanted to focus it on one thing, because there's so many morphogens. We want to focus on one to see first if we can see any changes with this specific one, and then possibly focus on other ones. So the reason why we chose that one is because we found other experiments that did work, and we want to see if we can do this for ourselves.
And so we knew that there is something going on, we just don't know exactly what, and so that's why we chose to do this specific experiment. Why?
Well, I mean, even the results just from a simulation, I mean, that is so exciting.
Yeah,
like,
exactly.
All right. So glucose is indicated by a strong yellow, urease magenta pink, and hydrogen sulfate by a black precipitate.
Right.
And so, as expected for our ground control sample, the machine was able to correctly identify our specimen as E. coli. And so the computer generates an antibiotic susceptibility pattern. These are the commonly used antibiotics. And once again, as expected, since K12 is a laboratory strain and non-pathogenic, it is pretty much susceptible to all the major antibiotics. Specifically, we like to highlight ampicillin and tetracycline.
This is our experimental sample that we sent to in conditions of microgravity.
Yep.
However, we were not able to retrieve the E. coli that we sent, but what overgrew it was Pseudomonas passim mobilis.
Oh, so you had contamination.
Yes.
How do you think that happened?
We were thinking maybe it was during the transport because it wasn't kept incubated. E. coli tends to grow resistance to harsh environments and conditions.
I tested coating an antibiotic in space, and I used the process of microencapsulation, which is when something is coated. You know how an M&M has the chocolate on the inside and the coating on the outside? That's encapsulation, but what I did was microencapsulation, which is way smaller. My hypothesis was that in space, the polyvinyl proteome would coat the drug more efficiently, and I mean efficiently by the drug would be coated more smoothly. There would be more of the coating on the drug, and that there would be less agglomeration.
What does that mean?
That less lumping together, basically sticking together.
Okay.
And I found in the supernatant from Earth that there was polymer that coated the Cipro. But from space, I just found like dried up cracks of the polymer.
Okay.
This tells me that there has to be agglomeration in the sediment. And what I found was, in Earth, it was coated evenly. And it was coated evenly because you can see the ciprofloxacin is stuck together. And there's holes in the middle, which means that the polymer, it focused directly at the cipro coating. But here, there was agglomeration because the polymer formed thick layers. So it didn't focus on the cipro like I expected it to. It just went by itself and formed thick layers. So basically, my results are that I found that in space, the cipro, which I thought would be coated more efficiently by the coating, was actually not coated as efficiently and was actually coated pretty poorly, you know.
Okay.
Overall, our resources mainly came from the internet. We also used local biologists, local scientists to be able to get our major information.
Alex, just as a last minute thing, said, why don't we do something with brine shrimp? And it ended up going all the way. We got to look in the microscope that took these pictures.
It was a computer screen that looked at these, and we got to magnify it. It was not a school microscope, it was like 10 times better.
The salinity that y'all had for the brine shrimp, did it match what like the salinity would have been for what their environment, I guess in marine,
we got asked that one. We tried to get as close as we can. The brine means salty, and they live in they grow in the salt lake.
Well, we had to add more salt because when we had the brine shrimp and the amount of water that we wanted, it wouldn't have filled the vial all the way. So we had to add more water because there couldn't be any leftover space. so therefore, if we added more water, we would have to add more salt.
But it does look like there was some development.
It kind of looked like it broke a little bit right here. So they might have started to hatch, but yeah, something might have just went completely wrong.
We also think when it starts to hatch, though, yeast goes right to it and repair. It kind of goes right to the opening. Next time.
They're like a do this, and we're gonna try to do their ISS, but we're gonna have some more challenges because it was more boys.
On this power slide, that's a picture of one of our cells, and if they grow arms which attach to each other, and these form myotubes, which form muscles, which form muscles. We got these from mice.
These are from our MDA unit that went up to space. We were able to take them out of the vials and put them on this little pastry type thing. And we were able to zoom in close enough to see that 3 out of 5 of them had developed.
Wow, nice.
Our experiment question was, how does microgravity affect the development of tilapia fish eggs?
Yes.
Our hypothesis was if eggs were sent into space, an environment with microgravity, then the parts of the fish may be in different locations than the one that didn't develop here on Earth. And our results were actually, surprisingly, none of the control embryos developed on Earth, but 3 out of 5 developed in space. Yeah, while we were researching for our tilapia project, we also found out an interesting fact that tilapia are mouth breeders. So here in the picture, you're able to see that they roll the fertilized eggs around in their mouth to prevent fungus from growing on the eggs. Now it's interesting because when we got our control MDA unit back, we used something called inoculating loops to put the scrapes of the eggs on petri dish. And it turns out that this is fungus and that this is bacteria. So it's weird how they're both different. But like control has fungus and bacteria, and in the experiment only has bacteria. But now that's weird. But like for the ones that were in space, these are both the same type. But we don't get why those two are the same from space, and the two in the control sample were different. We think that it has to do something with microgravity and the environment. Yeah.
So what we tested was the viability of Lactobacillus GG. Lactobacillus GG is a probiotic, which is a good bacteria, and it's found in dairy products like milk, cheese, and yogurt, etc. And it's also a bacteria in your intestines that keeps you healthy. And so it keeps your immune system up and running. And it was found in Russian cosmonaut research that the lactobacillus levels actually decreased over 96 days. And so we were wondering what exactly happened over those 96 days. So we thought that it was just the microgravity in general. And so we sent the bacteria up and just let it grow. And then we got our results back. And here on this graph, this is our Earth line, and then here is our space line. However, we did find out that it was the stress that in fact made the levels decrease, because we read an article that over time stress can decrease the levels of bacteria.
We tried to get crystals to help our friend Brittany. She has cancer and she's had it all her life. She has brain tumors and they've gotten so big they can't operate on them. She has about 1 more month left to live.
Oh my gosh.
And so the lysozyme could be used as a potential drug target for cancer.
Really? The protein crystals are temperature sensitive?
Yes.
Okay. And I take it then they're also moisture sensitive?
Not really, but they can be depending on what moisture it is.
Oh, okay. Can you tell me what this picture is?
Yes, that is one of our crystals during X-ray diffraction.
Which is how you would measure the crystals.
Yes.
Oh, okay. This is what you measure. That's how you measure the crystal, the crystalness of it, right?
Yeah.
Okay. Okay. And that wasn't one of your space samples, was it?
No, that was the ground truth that we did not do, but It was our lovely scientists that helped us did.
Okay.
We did the honey concentration for 10%, 5%, 1%, 0.5%, and 0.1% in the control. And those are the results we contained. We did 2 experiments so we can compare it. The first one was for the dark bacteria, so in the dark for 48 hours. And as the honey, that one's the blue one. And as the honey concentration increased, the bacteria just decreased. Now for the bacteria that was stored for 5 days in the dark, it decreased until it hit 1% of honey concentration. And as the honey started to increase, so did the bacteria. And we predict that that was because the bacteria just started to feed off the sugar that was in the honey. So that's kind of how we compared it to each other.
Alright, the osmotic effect of the high sugar content can be sufficient to inhibit microbial growth. The honey increases in dilution and the enzyme glucose oxidase and then is activated in water, resulting in the production of hydrogen oxide, which turns out to be toxic for bacteria.
The difference is that this one was washed with the colorizer. The way we need to wash it with the colorizer because since there's still a couple of salt particles in there, we want to wash away that so that the salts will be more visible.
EMB is a media that almost is specific to E. coli. The layer that the biofilm has created, you need to kind of break through that very gently to see the individual E. coli cells. You know they're in there. It is the biofilm that they are.
We ran them in the microscan automated microbial identification system, which is available in our labs. That has a database that compares them. But we also send them to where did we send them?
We did send them out to a lab that specializes in it just to verify and back up our results.
Acidic environment